By the middle of the 19th century, Gregor Mendel had unlocked the secrets of heredity. He showed that a trait like roundness in a pea did not blend with a wrinkled trait. Mendel proposed that factors, which we now call genes, survive intact from generation to generation and preserve roundness and wrinkledness in fixed mathematical proportion. Mendel explained how a trait, or phenotype, could suddenly reappear in a population. Because the genes which produce variations are hidden and survive from generation to generation, they occasionally produce a different phenotype. A change in the environment might favor one variation over another. Slowly, natural selection, acting on this phenotype, could bring about the evolution of the species. Mendel's work was a theoretical description of how variation might occur, but neither Mendel nor Darwin were aware of the actual mechanism which could produce variation. It would take the work of many cell biologists to reveal the actual mechanism of variation. Around the beginning of this century, scientists began to suspect that the chromosomes contain the genetic information which determines both the function and appearance of the organism. Here is what biologists observed. Prior to a cell's division, chromosomes within the nucleus would first replicate, line up, and then separate. Finally, one member of the replicate goes to each of the daughter cells. This process is called mitosis. While this method of cell reproduction guarantees continuity, mitosis certainly doesn't provide the variation necessary to fuel evolution. Possessing the identical genetic material of the parent, the offspring would remain mere copies of the parent. In terms of evolution, mitosis is a relatively blind end. It was nature's invention of sexual reproduction that enabled organisms to generate variability. When two gametes fuse during fertilization, each parent contributes its complementary half of the genetic information to the offspring. But first, there was a hurdle that nature had to overcome. All cells, except gametes, in sexually reproducing organisms are diploid. That is, they contain a set of chromosomes from each parent. We can represent this mathematically by assigning n to each set of chromosomes. If we start with two diploid gametes, an egg and a sperm, and each contributes its full complement of chromosomes, fertilization would produce a tetraploid offspring. And the next generation, an octoploid. Until we might imagine a cell overstuffed with chromosomes. If sexual reproduction were to be successful, the diploid organism needed to reduce its number of chromosomes as it divided. And how, then, does nature carry out the act? Let us suppose a hypothetical diploid cell contains both the dominant allele and recessive allele for a given trait. During the primary stages, the chromosomes replicate exactly and pair up. In the secondary stages, the chromosomes migrate in opposite directions, and two cells are formed. Finally, these two cells divide again, and this time, the duplicate chromosomes separate to form 4 haploid cells. These 4 haploid cells are gametes. And meiosis is the name given to the process we've just watched. In meiosis, a single diploid cell becomes 4 gametes. Now, when a female gamete combines with one of these male gametes, note how the diploid configuration is restored with a chromosome from each parent. That's a simple model of meiosis. Now consider a more likely diploid cell containing not one, but several pairs of chromosomes. Each of these pairs will, through meiosis, eventually contribute one chromosome to each of the 4 gametes. Now, which chromosomes from which pair will wind up together in the same gamete? Impossible to tell, because meiosis, like a giant slot machine, selects them by random assortment. Suppose each window represents a different chromosome pair, and each carries a different trait. Each trait will have an even chance of being recessive or dominant. Crank the random generator. The number of possible combinations is a stingy 8. Now, increase the number of chromosomes to 23. That's the number in a human gamete. Ah, the random assortment payoff is in excess of 8 million possible combinations of chromosomes in each gamete. But even this model of meiosis, with its staggering variation, is still an oversimplification, because nature is more complicated. Working once again with a single diploid pair, our model gives us not 4 different gametes, but 2 pairs of identical gametes. At this stage of meiosis, the chromosomes carry out a maneuver called crossover. Note how the chromosomes exchange material. What is important as we watch the cell perform its choreography is the net result of the crossover. Not 2, but 4 distinct haploids, even more variation. Crossover and random assortment produce mind-boggling variation. The human zygote has 46 chromosomes, and by conservative estimates, 10,000 genes on each chromosome. Because of meiosis, the number of potential genotypes the individual might take on at conception is astronomical, more than all the molecules in the universe. Though Darwin wasn't familiar with meiosis, his intuition was uncanny. Evolution, he postulated, was a result of these countless minor variations, all adding up over the eons. But what was the chemical basis of these countless variations? In 1953, Watson and Crick reported that chromosomes are made up of molecules called DNA, the now familiar double helix. Untwisted, it resembles a ladder, with rungs of 4 key nitrogenous bases: adenine, thymine, guanine, and cytosine. The sequence of these bases makes up the genetic code. During both meiosis and mitosis, the DNA ladder unzips. Each half of the ladder rebuilds its complementary side from chemicals in the organism. One DNA strand becomes 2 double helix molecules. This DNA replication is the foundation of meiosis, a way of reshuffling genes to produce constant variation.